Okay, I hope everyone is doing well and that you're all staying at home as best as you can, unless you absolutely have to go out. These are some crazy times we are living through, but always remember that we are in this together, and I hope that this class is still enjoyable and easy to understand. So you should have gotten an email with my plan of what the rest of the semester is going to be like. So you can always refer to that if you want to figure out what all is going to be going on for the rest of the semester. So this is the first recording for the lecture series for finishing up the semester. And we are finally getting into our animal unit. I'm going ahead and making a video of this first presentation because a lot of you did leave on Saturday, which I understand. So I did go ahead and make a video for this one. All right, so an overview of animal diversity. First, animals are multicellular, heterotrophic eukaryotes. So several traits that define animals are the nutritional mode. So we have up here that they're heterotrophic. Heterotrophic means that they have to consume to get energy. The second point is the cell structure and specialization. We're going to see a lot of different types of specialized cells depending on the animal clade, phyla, or class that we are talking about. And cell specialization. We're going to see a lot of complex tissues as we move through our evolutionary tree. The third point is reproduction and development. And we will go through different types of development as we go through this lecture. And I did want to put on here, of course, remember there are exceptions to every rule. Biology is not black and white. There are many shades of gray. And if you ever go online and start looking some stuff up, you will probably find things that contradict what I am teaching you. What I am teaching is the overall trend for these animal groups, but there's always going to be exceptions to these rules. So I would just always want to put that in there. All right, first, kingdom, Animalia. So we are going into a new kingdom. Animals are eukaryotic, so they have a true cell. That means they have a nucleus, a membrane-bound cell with organelles, and they are in the domain eukarya. Heterotrophic, so they eat, consume for energy. These are going to be organic compounds that they're consuming. They can respond to stimuli. So with our plant section, I talked about how they could respond to light, photoperiod, they could respond to moisture, but now we're actually going to see contractile tissue or muscle. So they can actually, it's a pretty fancy way of saying if you poke it, they'll respond to it. That's a generality, but more or less. Multicellular. So that means that they are composed of many cells, multicellular, many cells, organized with specialized cells. So we'll talk about different types of cells as we go through this as well. They don't have cell walls. This is really a big thing that separates them from plants and fungi. Tissues. So tissues, again, are groups of cells that serve for specific functions. The only animal phyla that does not have true tissues are sponges. Sponges are the most basal or least evolved member of the kingdom Animalia, and we will go over what they're composed of in the next lecture. Diploid dominant life cycle. So even human beings, we are diploid organisms. So each one of our cells that makes up of our skin, our liver, our stomach, our hair, they are diploid. They have 2 full sets of chromosomes per cell. This is the only part of us and of animals that are haploid are going to be the gametes, the egg and the sperm. And then we're going to see a bunch of different types of metamorphoses. So for metamorphosis, we're going to see terms like embryo, larva, juvenile to adult. So it's just the transition from each one of these life stages, while still remaining a diploid dominant organism. All right, so some differences between algae and plants, between animals. So first off, plants are going to be autotrophic, so they use light from the sun to produce simple sugars through photosynthesis. Animals, on the other hand, are heterotrophic, so they are consuming organic compounds. Cell walls versus no cell walls. The cell walls of plants are composed of cellulose. The cell walls of fungi are composed of chitin. Animals, on the other hand, they have no cell walls. They do still have a cell membrane, that phospholipid bilayer that you've probably heard all about in your Bio 100 classes. They still have that phospholipid bilayer composing the cell membrane. Plants have relatively nitrogen-poor tissue, while animals have nitrogen-rich tissue, especially if you look at muscle. Muscle has a lot of nitrogen. Remember, as I talked all through our plant series and we talked about the nitrogen cycles, how important nitrogen was to our plants. The fourth point, the plants are supported by cellulose and lignin, or if you're a fungus, then you're going to be supported by chitin. Animals, on the other hand, are supported by collagen, and collagen is that substance that composes our ears, our nose. It's a soft tissue, but it's pretty rigid. So you can bend it, like you can bend your ear forward, but it'll just spring right back into shape. Plants have a large external surface area. I talked all about why surface area and increasing it was important for plants. Animals, on the other hand, are going to have a small external surface area, so they're going to be conserving energy more. And the sixth point, algae and plants have relatively simple tissues, while animals are going to have complex tissues, and we're going to see these in the muscle system, nervous system, etc. All right, reproduction and development. The sexual reproduction is going to be the most common mode of reproduction. However, a lot of the organisms that we start talking about do exhibit different types of asexual reproduction. Post-fertilization, so this is after the fusion of egg and sperm, produces the zygote. That zygote is going to undergo rapid cellular division. So this GIF I have here is a rendering of what that would look like, that rapid cellular development of after egg and sperm have fused. This is called cleavage, and once that cellular division is done, it forms the blastula. After the blastula is formed, it will go through this process called gastrulation. So here we go through the whole process. Right after fertilization, we have our zygote. That zygote is going to undergo rapid cellular division called cleavage. So here is the early stages of cleavage. This is after cleavage is completed. It forms the blastula. And inside of the blastula, you can see this plane here. So we're kind of looking inside of the blastula, and it is hollow inside of the blastula. That opening, that empty space in there is called the blastosole. Now, for animals, there's going to be a process called gastrulation. So on one end of the blastula, we're going to have an indentation of that exterior wall, and that process of indentation is called gastrulation. So here's the definition of gastrulation. One end of the embryo folds inward, expands, and eventually fills the blastosole, producing layers of embryonic tissue. So here we start seeing our embryonic tissue. And we have some terms here that I'm going to introduce a little bit later in the lecture: endoderm, ectoderm, and when we get more advanced, the mesoderm. But we will get there in just a sec. So first, a little bit of history of animals. The kingdom Animalia includes a lot of diversity of living species, and an even greater diversity of extinct ones. So what we have on the planet now is just a fraction of the amount of animal diversity that has historically existed on the planet. The common ancestor of animals may have lived as much as 800 million years ago, so very, very, very long time. And it might have resembled a modern choanoflagellate. So that should sound familiar because that was one of the protists that we learned about. So this is what one of those choanoflagellates look like. So this is what we think, what scientists believe, looks like the closest relative to animals. And we're going to see cells that look very similar to this choanoflagellate in our sponges. And that's going to be in the next unit whenever we go into talking about sponges and what they're composed of, because again, they lack true tissues, but they are the oldest type of animal. So here is a cladogram. I'm going to be using a couple of different cladograms throughout this entire lecture series. This is one that'll keep popping up. So as we move from the bottom left to the top right, we are moving through evolutionary time. So we are becoming more and more derived. So that might sound familiar because as we move through evolutionary time, we pick up more and more derived traits. So if we are looking at these guys up here, we have starfish and we have this frog. And if we want to group those into a single clade, then the derived trait for this clade is going to be deuterostome development. So this is a new trait that is only found in this clade. Everything past the common ancestor, which is right here. Remember, this is called the node. The node represents the common ancestor. Everything past that point, going back through evolutionary time, going back through evolutionary time, these are our ancestral traits. So these are going to be inherited for all of the other organisms as we move through our cladogram. All right, now we're going to go over body plans. So scientists tend to categorize animals according to their morphological and developmental trait. This is how do they look, like what is their morphology, what do they look like, and how do they develop. So first we can look at different types of body plans. We're going to look at 3 different things in regards to body plans: symmetry, tissue composition, and body cavities. For symmetry, the first type of symmetry is going to be found in sponges. This is asymmetry, which actually means no symmetry. So for sponges, they have no distinct type of symmetry. You can cut them along any plane, and you will not have 2 equal sides. Next, we have radial symmetry. Radial symmetry is where you can cut along any plane of the organism. So here we have an anemone, and you can slice it along any plane and have 2 equal sides. The most advanced type of symmetry is bilateral symmetry. This is because when you become bilaterally symmetrical, you have 2 distinct ends. And when you have 2 distinct ends, you can start focusing the amount of sensory organs to one end of the body. This is called cephalization and will eventually result in the formation of a head and a brain. So the more advanced organisms are bilaterally symmetrical. So yeah, bilaterally, you can cut them along one plane and you have 2 equal sides. If you cut them this way, you would not have 2 equal sides. And it forms the head. All right, next for body plans, we're going to get into tissues. Animal body plans can also vary according to tissue arrangement. Is it on the outside, the inside, or in the middle? So here I have a picture of what we are going to be going over. We have diploblastic organisms and triploblastic organisms. Tissues are going to be specialized cells that are going to be isolated from other tissues. So we're going to have tissue specialization. And during development, we can have up to 3 germ layers that will give rise to these specialized tissues and organs of the animal embryo. So depending on whether or not it results from the ectoderm or the endoderm, it can give rise to different organs and tissue systems. And we're going to go over each one of these in detail now. So here we have diploblastic organisms, di for 2, and so it's safe to assume that they have 2 layers. Triploblastic organisms, tri for 3, have 3 different germ layers. Our first layer is called the ectoderm. This is the outer layer. So for our diploblastic organisms, they have this outer layer. This is the ectoderm. And here's the ectoderm for our triploblastic organisms. This is going to develop into things such as skin, sensory organs, and nerves. The middle layer is only found in our triploblastic organisms. Meso means middle. And the mesoderm can develop into things such as the skeleton, muscle, the circulatory system, and more advanced reproductive systems. Then finally, the last inner layer is called the endoderm. Endo for inside or within. So here is our endoderm for our diploblastic organisms. And here it is for our triploblastic organisms. And this is going to develop into the gut and digestive organs. All right, for body cavities are only going to be for our triploblastic organisms. A true body cavity is called a coelom. All triploblastic, so 3 germ layers, are classified based on the presence or absence of a body cavity. Animals can be classified on whether they have a true body cavity, and the older groups of animals will have none. So we have 3 different types: acoelomates, pseudocoelomates, and a true coelomate. And we're going to go over each one of these. So first for our acoelomates, again we have this A in front, which means without. So these organisms do not have a true body cavity. They have no coelom. So we do still have our 3 germ layers. We have our ectoderm in blue and mesoderm in the middle in red, and then the endoderm in yellow. What you don't see, though, is a cavity in here. Like, you might see this hole and think that that's a body cavity, but that is actually the digestive tube. So that does not count. That's not a body cavity. That's the digestive tract. Some examples of acoelomates are going to be marine flatworms, flukes, tapeworms. So pretty much this menagerie of organisms that you see here. Pseudocoelomates, this front word here, pseudo. Pseudo means false. So false cavity, a false coelom. The reason why we call this a false coelom, we have our germ layers. We have our ectoderm in blue. We have our mesoderm in red. And then we have our endoderm in yellow. And we do have this cavity here. So now we do have a cavity, but this is considered a false cavity because it is not completely surrounded by mesoderm. It's touching 2 different layers. It's touching the mesoderm in red, and it's touching the endoderm in yellow. So this is considered not a true body cavity. The reason why body cavities, even this false body cavity, are really important is because it can help with more directional type of movement. So whenever we get to start learning about our nematodes, so nematodes are a really great example of pseudocoelomates, and they also develop 2 different types of muscle. So we have, just type of muscle. They have muscle, and then they have this space here that is fluid filled, and that creates pressure within the body. So when you contract one side of the body, let's say this bows inward, contracting, then this will relax and expand. And when you combine that with the body pressure and that muscle movement, these nematodes can do a very sophisticated type of movement I like to refer to as wiggling. They are capable of wiggling, which previous organisms were not. Finally, we have our most advanced type of body cavity, coelomates. So coelomates mean they have a true body cavity. So if we look at this diagram, we have our ectoderm, our mesoderm in pink, and then our endoderm. And you can see here's our coelom, our true body cavity, and it is completely surrounded by that middle layer, the mesoderm. It's not touching any other germ layer. This makes the pressure inside the body of our coelomates a little bit more sophisticated. That muscle movement is going to have a couple different types of muscle, so you can have more advanced, more directional, and intentional types of movement. All right, finally, we have different types of development. Protostomes versus deuterostomes. Many animals are classified whether they are protostomes or if they are deuterostome type development. We see differences in cleavage, the coelom formation, and the blastopore development. So for the duration of these next few slides, I'm going to have protostomes on the left and deuterostomes on the right. So first, differences in cleavage for development. Protostome development, the cleavage is going to be spiraled in nature, indeterminate, which means it ends. So this is our protostome over here. Once these cells start to divide, it's going to be kind of in a spiral formation as it goes down. Our deuterostome development, on the other hand, cleavage is going to be radial, so it's going to start at one point and radiate down all at the same time. So it radiates down, radial. And it is indeterminate, so that can keep on dividing. Finally, we have differences in how the coelom is formed. So this is that body cavity. For protostomes, the splitting of solid masses of mesoderm forms the coelom. So here's our protostome over here. We have that indentation as a result of gastrulation. Once gastrulation is over, it forms this empty space inside, and we call this the archenteron. So for our protostomes, we just had some budding of the mesoderm, and it kind of closed itself off and creates its own little layer. So here is the coelom, as we're going to be making inside of the mesoderm. For our deuterostome, mesoderm buds from the wall of the archenteron to form the coelom. So instead of budding off from the mesoderm, it's actually going to bud off from the wall of the archenteron, again, this space here. Finally, differences in differences in the blastopore. The blastopore, again, is that first opening of the digestive tube that develops in the embryo. So the blastopore is either going to develop into the mouth or into the anus. There's two ends to the digestive system, and so there's two different types of development. For our protostomes, the blastopore develops into the mouth first. In our deuterostomes, the blastopore develops into the anus first. More advanced organisms, including human beings, are deuterostomes. We are in deuterostome. We have some differences in views of animal phylogeny. There's been new molecular data that's been coming out that rather being based on morphology and development, it's based on molecular data. So we're going to go over kind of the hypotheses and relationships of these animal groups. So here are our cladograms. So this is a pretty simple one. So this phylogeny at the top left, this is a phylogeny based on morphology and development. So this is what do they look like and how do they develop. So everything up until about this point remains the same. But once we get into bilateria, so bilateral organisms, we have two main groups, deuterostomes and protostomes. But if we look at the phylogeny based on molecular data, that protostome lineage is further separated into two different groups. So this is where our differences are located, and we're seeing it in our protostomes. Protostomes for our molecular data are being separated into these group Lophotrochozoa and Ectozoa. The points of agreement. So this is how do these two things, what do they agree on? That all animals share a common ancestor, that sponges are the most basal or least evolved group of the animal kingdom. Eumetazoans are a clade of animals that have true tissues. So we have this down here, true tissues. Most animal phyla belong to the clade bilaterians, so bilaterally symmetrical. Chordates and a few others all belong to the clade Deuterostomia, so they are deuterostomes. So there's some progress in resolving these relationships. So again, this is really where we are seeing our differences. That's the morphological data. This is the molecular data. Ectozoa is separated out and Lophotrochozoa is separated out for our molecular data. The reason behind this is that ectozoans, they all shed some kind of exoskeleton. This is a picture that I personally took from a research trip in Panama, and this is an insect called a cicada. Cicada live as larvae underground for the vast majority of their life, but once they're ready to become adults and reproduce, they pop up out of the ground, shed that exoskeleton, and then go through sexual reproduction. For the group Lophotrochozoa, these guys all share some similar feeding structure. These are called lophophores. So these are some examples of lophophores. It's this feathered fan-like structure that can be used for feeding. So we will see that many of these go through a distinct developmental stage called the trochophore larval stage. And so that's something that this group shares. All right, that finishes up the first section of this lecture series. So I hope this was understandable, and we will continue on the next video.